Before I move into this, I would like to share with you all that the third edition of the APS Rose Compendium is currently at the printer, should be available for purchase in December, and they expect to start advertising around Thanksgiving, so for pre-order. So just wanted to get that out of the way. So let me... So we have previously established colonies of Phylacoptes fructiferus in the lab and transferred those to the greenhouse. We've observed symptoms in 2 to 3 months after mite transfer using cages like this, or additional methods. So a second method was by just attaching a flower from an infected plant and just twisting that onto a shoot of a healthy plant, covering that in a plastic bag to maintain the mites and also maintain humidity, and then later transfer that to plastic cages in the greenhouse. Okay. Not wanting to move here. There we go. So these are symptoms from attaching a flower from a rose rosette positive plant, and started to see symptoms after about 10 weeks after flower attachment while the plants were still in the lab. These were, A here on the left was our initial cages, and this was too small an area of the 25 micron mesh necessary to keep the mites in to allow effective ventilation. The second generation of cages, we increased that area of mesh about 3 times. And then the third generation of cages, we added the larger area of mesh on opposite sides of the cage and in line with between the cooling pads on one side of the greenhouse and the fans pulling the air across the greenhouse on the other. So even with those third generation cages and with evaporative cooling and fans moving the cooled air across the greenhouses, the temperatures inside the cages sometimes reached greater than 110 degrees centigrade this summer. Sorry, Fahrenheit, that's a mistake. And mite colonies could not be maintained. The plants did okay, but the mites didn't. So we're therefore using leaf cages and flower reattachment to reinitiate mite colonies, some of which will be maintained in a growth chamber as a backup. And we are looking at cooling units to be able to provide cooled air into the greenhouse mite cages, because apparently there's sufficient solar heating inside those cages to raise the temperature considerably above the greenhouse temperature. Then we've also been grafting plants with rose rosette infected shoots to increase the number of rose rosette plants that we can maintain in the greenhouse for use in grafting to challenge the rose corset. We received all of those plants in July, and we're going to need lots of multiple small diameter infected shoots for grafting to those core genotypes, as some of those have much more slender stems than the knockout and some of the other varieties. So we've had some setbacks, but we're now making progress. We've mentioned before that to simplify grafting efficiently, we designed and printed, 3D printed a grafting jig to make uniform cuts at a same angle across stems of both the stalk and scion to ensure a similar vascular tissue profile and the same area of on both sides in order to make a strong graft union. And then we've also 3D printed cages to enclose mites on a detached flowering stem using a soft foam gasket to seal around the stem. These cages were designed, the two halves are identical and they just click seal together. So on the top left here, you'll see a variety of angles of the 3D printed grafting jig. This is essentially about 1.5 inches long. There's a 17.5-degree angle across there, which gives a good surface area of the graft where the tissue can knit together. And then because we're making the same, using shoots of the same diameter and cutting a uniform angle, we get good contact between the vascular system and the pith, so that we have a pretty good grafting success. At the bottom on the left, you can see the two halves of the cage assembled as seen from the base. And then here we have the separate halves looking from the top of the cage. So those two little holes like nostrils there are for putting a string or wire through to support the cage once it's assembled over the rose flower. And then on the right, you can see a rose flower inside the cage and the stem of the rose in a preservative solution so that they can be maintained for, we hope, at least 2 weeks, so that we can do the, calculate the reproduction rate of the mites on the different rose genotypes, and also look at the number of feeding sites via scanning electron microscopy. Naranabat came to visit us in March. We showed him the leaf cages and the flower attachment to aid mite transmission. Demonstrated grafting for rose rosette transmission without the necessity for mites. And then he talked with Ron quite a while, looked at light microscopy and tabletop scanning electron microscopy for morphological mite identification. Then in September, Na sent his 2 graduate students, Alina Pakrel and Deepika Sharma, to spend a couple of days with us, with me, for use of the leaf cages and flower attachment for mite transmission and grafting for transmission without mites. And then with Ron, light and scanning electron microscopy for mite identification and the preparation of permanent slide mounts, etc. So we enjoyed having them visit us, and these are actually some of the roses that were graft inoculated by the 2 of them. On the left here, you can see 2 arrows out of 3 grafts that were made on this plant. This has actually only just recently been taken out of the bag that was maintaining humidity for the grafts. In the middle, there's concentration on 1 graft made by Alina, and on the right here, another made by Deepika, both of them surviving. So this is now what, 6 or 7 weeks since they're done, and they're still looking good. And I hope that they are among the audience to see that. And then Andrew Jansen recently found out that mite autofluorescence enabled visualization of mites against a complex background. This was done using an axiozoom fluorescence microscope, which has wide field as well as much narrower capabilities. And Austin Fife has now utilized it. So that was with a different mite and host species. But Austin Fife just applied that to Phylacoptes fructifilus on rose sepals. And rose sepals is the preferred location for the mites to feed on the plants. The sepals typically have a very dense growth of trichomes, making it quite difficult to see the mites because the mites are typically hiding in and among the trichomes. and showed that the mites can easily be observed among the trichomes when using appropriate illumination and filters. And this will hopefully be a far easier method of determining the mite numbers on the sepals of the core set of rose genotypes to compare their longevity and reproductive rate, although we'll still need scanning electron microscopy imaging to examine the numbers of feeding sites per mite that's present. But most of you have probably seen this image on the left before and on the right. But if you look at the image on the left, the original scanning electron microscope image, I don't know how many of you would be able to pick out the mites without looking at the colorized image on the right. And in the colorized image, the trichomes and eggs of the mites are shown in white. The mites are shown and spermatophores are shown in this amber orange color. So here is a mite. Here are some spermatophores. There are both adults and nymphal stages of the mites present there. Then here the red coloration is the heads of glandular trichomes, and here the columnar stems of the trichomes, of glandular trichomes, are also in a gray color. So that's just what we're able to do with scanning electron microscopy. So you can see from the left-hand image, it's difficult to pick out and count the mites. So the autofluorescence is hopefully going to make that very much easier for us. So here on the left, you see an axiozoom image under white light, where you can see the density of the trichomes on a rose sepal. And there is one mite there shown by this yellow arrow, but there's very poor contrast against the high trichome density. And this mite happens to be on the surface of the trichomes. Frequently, you find them more around the margins of the, or at least they're easier to find around the margins of the sepal where the trichomes are less dense, but they can also be found deep among those trichomes as shown in the previous SEM image. So the panel on the right here is a higher magnification of that same mite, arrow here. This is shown under lighting conditions similar for those used for GFP detection, showing a higher contrast of the mite relative to white light. I mean, it's fairly easy to pick out that mite. There are obviously trichomes in the background, but look at the density of trichomes here. You're really hardly seeing them here at all. And Austin is currently examining different lighting and filter selections to further improve the detection of the area-fired mites. So my thanks to all of the Beltsville group for their roles in this work. Ron is the mite expert. Andrew is a microscopist, as is Austin Fife, and doing the SEM and light microscopy work for us. And then Ramon has been working with me on the, well, actually Ramon takes the lead on the rose rosette detection, and I'm doing the grafting and the mite transmission. So any questions?
Thank you, John. Any questions for John?